Well, I'm excited. I don't know about you, but I am excited. We've got exam 2 out of the way, and now we're going to wrap up the glucose metabolism story. This story that we started at the beginning of Unit 2, we are now wrapping up at the beginning of the Unit 3. This is such a big story that it spans across more than an entire unit. I'm also excited because of, as I shared with you in my recent Canvas announcement, the light's at the end of the tunnel. Kudos to every single one of you for working as hard and as consistently as you have worked. We've all done more than we've ever thought we would have to do in a single academic year. My hat goes off to each and every one of you for the grit and the resilience you've shown. And like I said, we're almost there. We're almost at the finish line. This is also the beginning of the third from last week of the course. I'm going to be completely honest and forthright with you. Next week sucks. That's the blunt truth of it. It's very, very dry and heavy biochemistry. We talk about amino acid biosynthesis, a little bit of nitrogenous base biosynthesis. Not my favorite week at all. In fact, my least favorite week of the entire course, but we have to cover it. For those of you that are going into healthcare, for those of you that intend to go on to graduate school, the expectation is going to be there that you know something about amino acid and nitrogenous base metabolism, so we have to cover it. And then we end with photosynthesis, which many students think of as in anticipation of it, they think it's going to be the worst week of the semester. It's actually the best week of the semester. Photosynthesis allows us to take everything we've been learning about all semester and condense it down into one topic. Now, I don't mean to make it sound complex or dense. We just pull from everything else we've learned. So it's like a capstone experience to the course. I really have tons of fun teaching photosynthesis, and I think we're going to finish this semester strong. So great week right now. Finish up the glucose story, talk about all this amazing stuff of the ETC and oxidative phosphorylation. Very sucky week next week, to be completely honest with it, and then we end strong with photosynthesis. So let's dive right in here. Based on your comments, it really came down to, it sounded like the class was hungry for the same kind of thing we did for glycolysis in the citric acid cycle. Context, big picture. Get more linear, tell the story, strip away the fat, and so that's what we're going to do here. I do want to point out that another topic that came up quite commonly in the reflections from the Edpuzzle videos was the electron potentials. The whole idea of positive delta E's give negative delta G's, and how to determine what's going to be oxidized and what's going to be reduced in a redox reaction. I hear you loud and clear. I know that needs review, but that's going to be embedded in the challenge question. So in the challenge question video, we're going to touch on and review all of that there. I really do want to focus this video on the nuts and bolts of the end of glucose metabolism. So rest assured you're going to get reviews on redox reactions and electron potentials, but just not right here. Okay. So what you've been staring at this whole time is, of course, a very, very crude, misshapen mitochondria. That's really what this is meant to show. This is the mitochondrial membrane, the inner membrane, this is the inner membrane space, and this is the matrix of the mitochondria. Glycolysis occurred all out here. Glycolysis is cytoplasmic. So glucose is transported into the cell through a glucose transporter, and glycolysis and pyruvate production occurs here. And then pyruvate is transported into the matrix of the mitochondria, and it is converted into acetyl-CoA here in the matrix, and the citric acid cycle occurs here in the matrix. So we talked about glycolysis, the PDC, and the citric acid cycle, just to give us context. Now, the end result of the citric acid cycle, right here in the matrix, is a lot of NADH plus H. In fact, we have 4 individually reduced NADH plus H's. One of those comes from the PDC. One of those comes from our first oxidative phosphorylation in step 3 of the citric acid cycle, step 4 of the citric acid cycle, and step 8 of the citric acid cycle. That is where we've gotten these 8 pairs of electrons, these 8 pairs of protons. We also have one more pair of electrons and protons from the citric acid cycle, but they are not on a mobile electron carrier. They are stuck on FADH2. And more importantly, they are stuck on succinate dehydrogenase. The succinate dehydrogenase catalyzes the conversion of succinate to fumarate in the citric acid cycle. So what is that? That's going to be step 6. And we said in that citric acid cycle lecture that we would see those electrons again. And they're embedded here in the membrane. Succinate dehydrogenase is a inner membrane protein of the mitochondria. So as I've drawn it here, it's embedded in that inner membrane and it's still carrying FADH2. So those are our 10 electrons and those are our 10 protons. And that's the end result of everything we've talked about so far. So this brings us to the end of Unit 2. So let's dive in here to the electron transport chain and talk about it in this context. Pick up the story right where we left off. So the first complex of the electron transport chain is complex 1. That's its easy to remember name, complex 1. It is more officially and probably more aptly referred to as NADH CoQ oxidoreductase. The reason why I favor that name is because now I don't have to memorize anything but that name. That name tells me everything. NADH CoQ oxidoreductase. This thing is going to oxidize NADH. It's going to reduce CoQ. That's its job. So, these electrons and protons from NADH plus H individually, all of them, but individually, not all at once, one at a time, they're going to offload their electrons and protons to complex 1. More specifically, there's a cofactor in complex 1 called FMN. It's a flavin, and FMN is going to be reduced to FMNH2. So FMN is accepting the electrons and protons from NAD. NADH plus H gets oxidized to NAD+, and it passes its electrons and protons to FMNH2. FMN becomes reduced. FMNH2 is going to pass those electrons to these iron-sulfur clusters, but only the electrons. The protons from FMNH2, and those are the protons here from NAD, the protons are going to be pumped into the inner membrane space. So key point here, now we've added protons to the inner membrane space, we've only passed the electrons to the iron-sulfur clusters. The iron-sulfur clusters are going to pass those electrons to coenzyme Q, or CoQ. Remember, it's NADH CoQ oxidoreductase. CoQ is going to be reduced. So the electrons get passed from those iron-sulfur clusters to CoQ. CoQ, when it's reduced, becomes CoQH2, but these protons are long gone. So these two protons, they came from this matrix of the mitochondria. You know from the lectures what we're ultimately creating here is a concentration gradient. What we want to wind up with is a lot more proton in the inner membrane space and a lot less proton in the matrix of the mitochondria. And we're contributing to that in two ways. We're putting protons into the inner membrane space, and we're absorbing protons from the matrix. We're doing both. So let's continue this chain of the electron transport chain, and then we'll come back to our friend at the end. So now the electrons from NADH2 are on CoQ. The electrons went from NADH2 to FMN, to iron-sulfur clusters to CoQ. The protons took a little bit less of a linear path. The protons went from NADH plus H to FMN, and then into the inner membrane space, and new protons came in to help reduce CoQ. The CoQ is now reduced. CoQ is going to, I guess I'll switch my color. CoQ is going to offload those electrons to complex 3. Now, complex 3 also has a more official name. It's CoQ cytochrome C oxidoreductase. Love that. CoQ cytochrome C oxidoreductase. This is a complex that's going to oxidize CoQ, and it's going to reduce cytochrome C. Now, when CoQH2 offloads its electrons to complex 3, it's going to offload those electrons, but not the protons. The protons from from CoQ are going to leave CoQ as it oxidizes and enter the inner membrane space. Again, we're contributing to that concentration gradient. Only the electrons will be transferred to this complex 3. In this complex 3 are different cytochromes. Cytochromes are coenzymes that carry electrons, but only electrons. They don't carry protons. And we don't really talk too much about the details of what happens in complex 3, in CoQ's cytochrome C oxidoreductase. We only say that there's a number of different cytochromes in there, and the electrons kind of get passed between them all. That's sufficient for our class. Those electrons, because remember, that's all we're dealing with now. The protons are gone. Those electrons are going to be passed to another mobile electron carrier called cytochrome C. Cytochrome C is almost like our counterpoint of CoQ. Cytochrome C is going to carry electrons from one complex to another. And the complex that cytochrome C is going to carry these electrons to is, not surprisingly, complex 4. Complex 4's more formal name is cytochrome C oxidase. What does that tell us? That tells us these electrons aren't really going anywhere next. This is the end of the line. This isn't oxidoreductase. This is just cytochrome C oxidase. Cytochrome C is going to be oxidized, and that's pretty much it. The electrons are going to be passed from cytochrome C, one at a time, to copper, to a copper-containing compound in complex 4. Copper is going to transfer those electrons to cytochrome A, and cytochrome A is going to offload those electrons to oxygen. Oh, what do I have? I'll use blue. Oh, blue makes sense, actually. To oxygen. Let me put that down here. So the electrons are being passed to oxygen. Now, as the electrons are being passed, let me see if I can focus this a little more. Oxygen is going to require protons to be fully reduced. So we are going to get protons from the matrix to fully reduce oxygen. These 4 protons are going to join this oxygen and make 2 water molecules. So electrons one at a time are going to be added to O2. Electron 1. Now O2 is carrying an electron. Electron 2. Now O2 is carrying 2 electrons. Electron 3. O2 is carrying 3. Electron 4. Now those oxygens, they have 4 unpaired electrons between them. That's not good for oxygen. So oxygen is going to absorb these 4 protons from the matrix and make 2 molecules of water. And that water is released as a waste product. Not in urine, but in breath. So oxygen is really the garbage can of energy-spent electrons. These electrons are high power. These are the electrons we harvested from glucose. These are the electrons that we use the citric acid cycle to capture. The same electrons are ending up on oxygen, but they ain't high-powered anymore. Why? Because we've used all that power to pump all these protons. Every single transfer of these electrons, every single one of these redox reactions has released some energy from those electrons, leaving them spent. They're now garbage. So those dead, empty, energy-poor electrons are put onto oxygen. Oxygen is reduced to water, and that water is released as waste. So we even dump the electrons themselves as waste. We exhale them as the water vapor in our breath. Okay, so let's track that path one more time as a comprehensive story. Electrons that were harvested by the citric acid cycle on the mobile electron carriers NADH plus H arrive at complex 1. NADH plus H is oxidized to NAD+. That NAD+ goes back to the citric acid cycle to get more electrons. The protons and the electrons join up with FMN of complex 1, reducing it to FMNH2. FMN is oxidized and passes its electrons to iron-sulfur clusters, but iron-sulfur clusters can't accept the protons. The protons get pumped to the inner membrane space. The electrons are then passed to the mobile electron carrier CoQ, but CoQ, to be fully reduced, it does need protons, and it takes those protons from the matrix of the mitochondria, making that matrix more proton poor, which is what we want. Again, CoQ is a mobile electron carrier, so it's going to shuttle and scoot across the inner membrane to complex 3. It's going to offload its electrons to cytochromes in complex 3, but the protons can't be accepted by those cytochromes, so the protons are pumped across the inner membrane into the inner membrane space. Those electrons from glucose are now on cytochromes, and they're passed between different cytochromes within complex 3. At the end of that passing, those electrons are passed on to cytochrome C. Cytochrome C is a mobile electron carrier, and it's going to carry those electrons to complex 4 and dump them onto copper in complex 4. Copper is going to pass those electrons to cytochrome A, and cytochrome A is going to pass those now energy-spent electrons onto oxygen to reduce oxygen. But oxygen can't be reduced by electrons alone. Oxygen needs protons to be fully reduced to water, and it's going to get those protons from the matrix of the mitochondria. So we are both absorbing protons from the matrix and releasing or pumping protons into the inner membrane space, giving us that gradient. Many more protons in the inner membrane space, very few protons in the matrix. But we're not done, because we never talked about those 2 electrons and protons that are stuck on succinate dehydrogenase. Now, succinate dehydrogenase also has a second name. Its second name is complex 2 of the electron transport chain. And succinate dehydrogenase will pass its electrons and protons onto CoQ. That will oxidize FADH2 back to FAD+, and CoQ will be reduced to CoQH2, and then they'll follow the same path as the other electrons. So I'm taking up a lot of room here, but I like to think of the electron transport chain, well, it's not that I like to think about it. It is proper to think about the electron transport chain as kind of a fork in the road. It's complex 1 or complex 2 to complex 3 to complex 4. That's the true electron transport chain. These are all the electrons from the citric acid cycle that are on NAD, and these are all the electrons from the citric acid cycle that are on FAD, and they converge on CoQ, and CoQ brings those electrons to complex 3. So that is the first lecture in its barest bones nutshell of what's going on here. I love it. I mean, it's amazing to me how it all comes together, right? Glucose is converted into pyruvate. We make some ATP along the way, but we wanted pyruvate. Pyruvate's converted into acetyl-CoA, but we get some electrons and protons as well. Acetyl-CoA is completely stripped of electrons and protons as it goes through the citric acid cycle, giving us these, and these electrons and protons. And now we finally see what those electrons and protons have done. What have they done? They've created a proton gradient. That's all they've done. There's a lot of protons in the inner membrane space and not so many protons in the matrix. That brings us to the second lecture of the week. What happens now? We've got all these protons in the inner membrane space, and we're proton poor here in the matrix. And so what's going to happen? Well, also in this membrane, embedded in the membrane itself, is ATP synthase. It is this beautiful enzyme that is coupled. Within the enzyme itself, it's coupled. It is a proton channel, and that's what we see here. Protons are going to follow their gradient. They're going to go from the inner membrane space to the matrix. That's why we were building this gradient here. We pumped all these protons out into the inner membrane space here to create this gradient so that protons could flow back through ATP synthase. But as protons flow through ATP synthase spins like a turbine. And when it spins, it drives these different conformations of the head of the enzyme. So down here, what's going through the membrane itself is the proton channel. This is what spins, but the spinning of this proton channel is going to drive conformational changes in this head group here. So let me kind of just zoom that in a bit, if I would. I'm going to get rid of the citric acids, of the electron transport chain stuff, and I'm just going to draw what I just drew a little bit bigger, if I can. So we're going to do the side view like we just had it, and then we'll do the top view. So here is the proton channel. And here are the head groups. So I'm just drawing what I had before, big. This is the matrix of the mitochondria, the inner membrane space. Lots of protons here, fewer protons here, and protons are going to flow through this channel, driving the conformational change here. Now, what I want to do is look at this from the top. So if we imagine that this is the perspective we have here, we're looking at ATP synthase from the side, I'm going to now draw ATP synthase like this. So we're going to look down on it from the top. See if I can do that well enough. I need that to be bigger. This is only 3. So that's about a third, I hope. And that's about a third, I hope. It's not to scale, but it's good enough. So again, to give us some perspective, this is ATP synthase from the side, and this is ATP synthase from the top. And right in the center here is this little kind of protuberance. There is this shaft or protein pole that goes from the channel all the way to the tippy top of the head group. And as this spins, this channel spins, as protons go through it and it spins, this is going to spin as well. Now, what does this matter? Why does this matter? We did all of this to spin a protein? Like, is that really what we're saying? We converted glucose into pyruvate, pyruvate into acetyl-CoA, acetyl-CoA into electrons and protons and carbon dioxide, electrons and protons to the ETC. We did all of that. We created this proton gradient to make one protein spin? Really? Well, yeah, that's really why we did this. It was all down to this proton gradient, all down to the spinning of ATP synthase. Now, the head group of ATP synthase is made up of 3 domains, like I have here, and all 3 of those domains are always in 3 different conformations at any one time. And those conformations are referred to as loose, tight, and open. The loose conformation is going to bind independently ADP, adenosine diphosphate, and inorganic phosphate. The tight form is going to squeeze those together. Now, what do we mean by squeeze those together? We know what we mean. This is an enzyme. It's going to make transition state. It's going to create a scenario such that we can overcome that activation energy. We're going to drive transition state in the tight form. We're going to squeeze and pull and bang on these substrates until they're driven to form product, which is ATP. And then the open form is just that. It's open. That ATP that we just made is released. Loose, tight, open. Loose, bind ADP and phosphate. Tight, squeeze them into ATP. Open, release the ATP you just made. That's what each of these conformations does. But now let's spin this proton channel. So the proton channel spins because protons are going through it. As protons go through this channel, this spins. That's why we made the gradient. But as this spins, everything is going to change by one orientation. So over here I had loose a moment ago. That's going to become tight. Over here I had open. That's going to become loose. And over here I had tight. That's going to become open. Well, I shouldn't have erased it. What I had drawn here before was what bound to the loose form, ADP and inorganic phosphate. But now that this is tight, this is going to be squeezed together into ATP. The ATP was already made here when this was in the tight form a moment ago, but now that we've spun, this is now open. This ATP is going to be released. And what happens in the loose form? In the loose form, we get ADP and phosphate bound independently. Pretty cool, pretty nice. Now let's spin again. More protons go through the channel. We have that stalk spin. We have that protuberance occur. We have conformational changes. Let's spin again in the clockwise direction. This is going to become open. This is going to become tight. This is going to become loose. Well, what does the open form do? It releases the ATP that was just made. What was the tight form doing? It squeezes ADP and phosphate into ATP. What does the loose form do? It binds a new round of ADP and phosphate. Wonderful. Let's spin again. More protons go through the channel. Let's spin again. So this is going to become tight, this is going to become loose, this is going to become open. We take this ATP, we release it to the environment, we squeeze this ADP and phosphate, we make more ATP, and loose is going to be binding new and fresh substrate. And we just keep doing that. Look already. 1, 2, 3, 4, 5. We've already made 5 ATPs. We've only spun this thing a couple times. This keeps happening again and again and again and again. Loose, tight, open, loose, tight, open. ADP and phosphate. ATP release. ADP and phosphate. ATP release. You're doing that again and again. What's driving this massive output of ATP? Spinning. This spinning. That's all that's driving this massive output of ATP. Well, why is this spinning? Because there are protons to cycle through it. Why are there so many protons out here and so few protons? Why is that concentration gradient so extreme to drive this spinning, which drives this ATP production? Because of the electron transport chain. The electron transport chain is driving this concentration gradient. Well, what's giving the energy for the electron transport chain to create this gradient? High-powered electrons. Where do those high-powered electrons come from? Citric acid cycle. How did the citric acid cycle get anything to get high-powered electrons off of? Acetyl-CoA. Where did the acetyl-CoA come from? The PDC. What's the raw material for the PDC? Pyruvate. Where did that pyruvate come from? Glycolysis. And what was the input to glycolysis? Glucose that you consumed in your diet. Ah! I love it! Amazing. Last bit of the story. You can imagine that if this continues on and on and on, I mean, concentration gradients are not forever, right? Ultimately, you're going to reach a case where protons have gone through, and you're going to reach an equilibrium, either balanced protons or maybe even more protons out here in the inner membrane space than in the matrix. That's never going to happen in a healthy cell. Because as these protons are going through ATP synthase, and as that concentration gradient's being diminished, the electron transport chain is still in action. All of these things are happening simultaneously. So these are the protons that cytochrome C, that CoQ is absorbing as it becomes CoQH2. These are the protons that oxygen is absorbing as it becomes reduced to water. New protons are being put in the matrix as they're being offloaded by FMN. So we're continually generating and maintaining this concentration gradient because we are continuously absorbing protons out here by CoQ and oxygen, putting new protons in here by FMN, and those protons came from NADH plus H. And so this concentration gradient is constantly maintained. It's constantly present to drive the conformational change. What a lovely story. What an amazing story. Oh, I told you I was excited, and I didn't think I'd get this animated. I don't even know. I'm all a Twitter here. This is great stuff. So hopefully you find it just as interesting as I did. That's really the story. I mean, we've come full circle. All the ATP I've been promising you we would make, we just made it. And we make it by the bucketful as we just keep ATP synthase spinning, because we keep these protons flowing, because we keep the ETC running, because the citric acid cycle's always going, because the PDC is always active, because pyruvate's always being made by glycolysis, because we're always consuming sugars and glucose in our diet. Hopefully that makes more sense. Like I said, some of the other nuts and bolts things and the critical thinking around this we'll get in the challenge questions. I don't care that it's a short review video. I think it's more important to just get the take-home message here, and hopefully that's what happened. If not, if there's anything that's still confusing you about this, or if you just share my enthusiasm and you think that this is maybe even just 10% as awesome as I clearly think it is, let me know in the dialogue box that's going to pop up right now.